Everybody, it's Miss Peachy from your WCA Biology A class, and we are going to be looking at Unit 5, Lesson 4, 5, and 6 today, which are the lessons on your photosynthesis portfolio. So in this portfolio, you're going to be designing and completing a lab where you're demonstrating photosynthesis. And I just wanted to make sure I made this video for you because I want to give you some ideas on what you need to do for this and also explain some of the terms that you're going to be using. So just as a reminder, photosynthesis is a chemical process that plants can do to be able to take carbon dioxide and water combined with sunlight to create sugar. And sugar is food. It's a way in which plants can get energy. They do release oxygen in this process as well, but they don't use that oxygen. The oxygen is just something that is a byproduct of photosynthesis. So in this experiment, you are going to be designing a hypothesis, and you are going to be conducting your experiment and then looking at the results of that experiment. So I want to go through a couple of the key terms before we start. So your hypothesis is the answer that is thought to be correct for the observations. So when you make your hypothesis, you are going to be creating an if, then, because statement. If I do A, then I think B will happen, and this is why I think that. So that's how you should kind of formulate that hypothesis. Now, as you go through your experiment, you might find that your hypothesis is incorrect. Maybe your data does not support your hypothesis. So if you notice a difference between the hypothesis that you've made and what actually is happening, you can revise your hypothesis and come up with an alternative hypothesis. Or if you find that what you've done is completely off base, that there's no support for it whatsoever, there's absolutely no relationship here between the variables you're testing and the results that you're seeing, you can state that your hypothesis is a null hypothesis. This means that this hypothesis was completely wrong. It's not even something you want to revise, it's just completely wrong. When you are doing an experiment, it is completely okay to be wrong. In fact, in science, you're more oftentimes going to be wrong than you are going to be right. So if you end up with a null hypothesis or an alternative hypothesis instead of one that is supported, don't panic. That's completely normal. That's kind of what science is. A lot of it can be trial and error in a lot of ways. When you're designing your experiment, you also need to have a control for your experiment. The experimental control is the part of the experiment in which the variable is not changed. So we want to be able to see what would have happened if we didn't change anything at all. And all of these steps that we do they're part of the scientific method, which is a way that we can keep consistency from one experiment to the next, right? We're all kind of following the same procedure, the same general method in which we conduct science. There's definitely more accountability for that process, and it allows us to find patterns and observations and be objective when we're doing that kind of stuff. So I'm going to go on to lesson 5 here in this particular unit, and a couple more key terms that I want to define for you all. So we have independent variable and we have dependent variable, and we have, we've talked about these in the past in physical science last year. An independent variable is also called the manipulated variable. So I like to use that term as well. These are variables that are changed on purpose by you, the experimenter, in order to see how things change. It is what we are manipulating in the experiment. That's why we call it the manipulated variable. The dependent variable are variables that change because of what you've done. These are things that happen as a result of your independent variable. They are oftentimes called the responding variable because they respond to the manipulated variable. So an example would be, if I want to see, I grew tomatoes in my garden this summer, and if I really wanted to see how I could get the biggest and ripest fruit, the best tomato plants or the best tomatoes, I could play with the type of fertilizer that I'm using on my garden. So I'm going to try like 4 different brands of fertilizer, and I'm also going to have my control, which is no fertilizer at all. And then I'm going to see like how big my plants get, but more specifically, I want to see how big my tomatoes get and how many tomatoes I can produce, right? So the independent variable in that situation is the fertilizer, because I'm changing that from one trial to the next. The dependent variable is how big my plants get, how big my tomatoes get, how many tomatoes I'm able to grow. That's my dependent variable, the tomatoes themselves, right? So the size of the tomatoes, the deliciousness of the tomatoes, how many tomatoes are in each plant is a direct result of what kind of fertilizer I've applied. That brings us to quantitative and qualitative data. Quantitative data are things like numbers. I want to count the number of tomato plants per tomato vine. I also want to know how big those tomatoes are, like big tomatoes, nice big tomatoes. So maybe I can measure the length and the width and the height of them and get the volume of my tomatoes. But that's quantitative data. Qualitative data are data that you can get that you really can't quantify in numbers. How tasty those tomatoes are, right? They can be nice and big and round and juicy, but if they're really gross tasting, then I don't want to eat them. So the taste of the tomatoes is definitely important data, but it's not something I can describe numerically. Also things like the color of the tomatoes, the smell of the tomatoes, how juicy the tomatoes are, all those things that are kind of beyond the ability of us to describe in numbers are still important and still data that I want to make sure that I jot down and have that as relevant data. And then finally, replicable. The experiment needs to be something I can repeat and get the same results over and over again. I can do this experiment multiple times, or I could have multiple plants for each type of fertilizer. So maybe I have 5 plants that have type A fertilizer and 5 plants that have type B fertilizer. The more plants that I have participating in each type of fertilizer, the more kind of believable and repeatable my results actually are. So this is going to bring me to, I'm going to get to the course outline again and go to lesson 6. So here's your actual portfolio that you're going to submit. Now in the lesson itself, it asks you to just design this on your own, and you can look stuff up and you can find things online that might help you, but I decided it might be easier for me to kind of give you a template to follow. So this is going to be on my website by unit 5 lesson 4 through 6, it says lab worksheet. And so this is what you're going to turn in to me. So your lab is entitled What Factors Affect Photosynthesis? And we're going to start with something that we know. And we know that if I take a leaf and I submerge it in water and expose it to light, bubbles of oxygen will actually form. So I'm going to show you this, maybe. I just gotta find the right window. I always lose my windows here. Here it is. Okay. So I'm just going to play this. No, this doesn't. Oh, I see. Okay. Maybe I won't. Here we go. So as you can see here, and I'm just going to pause the screen, but as you can see on the left, this is our control. This is the glass that has no leaf in it. And this is our glass with a leaf in it. And as it sits for many, many minutes, we will notice bubbles of oxygen forming around the leaf, right? And so that's kind of the information that we're starting with here. That I know that if I put a leaf in water and put it in sunlight, the bubbles of oxygen will form. So you can kind of use that as your control in a way. That's kind of the standard. We know that's going to happen. So then there are some variables that you want to play with to see if that's going to change the situation. Does the type of leaf matter? Can we compare multiple different kinds of leaves to see if, like a spinach leaf versus a maple tree leaf versus a leaf on a flower, versus a lettuce leaf or something, can we experiment with different types of leaves, and see if you get more or less bubbles, right? So that's something you can experiment with. Does the color of the light matter? Maybe at home you've got some cool cellophane or some different like little pieces of plastic that are different colors, and you can put them on your light source and you can change the color of the light. You know, it doesn't matter if I'm using white light versus just blue light or green light or red light. So can I do this experiment with different colors of light and see if that changes the number or the amount of oxygen bubbles that I'm going to get? Does the intensity of the light matter? Can I, one second here. Sorry about that, guys. Can I actually have some of my leaf, I could put it in bright sunlight versus maybe I can put it into like a shady area versus artificial light, maybe a lamp on a table? Can I have at least 3 different types of light sources and see if that makes a difference in the number of bubbles that I see? Does the temperature of the water matter? Maybe if I use cold water versus lukewarm water versus hot water, will that change the number of bubbles that I see? And those are just some examples that I came up with. If you can think of another type of variable that you would like to test, please let me know, and we can kind of discuss whether or not that would work for this experiment. So you're going to be doing this. You're actually going to be gathering leaves, and you're going to be carrying out this experiment. The only materials that you need are leaves. They've got to be alive, so picked from a tree or a plant or something like that. Or like I said, you can take stuff from your refrigerator if you have like spinach or lettuce or something in your refrigerator. You need to have some sort of a clear container, a glass or a jar or something that you can make observations through. Water, something to kind of hold the leaf down under the water. So they suggest putting a little rock on that leaf so that it stays submersed. But you know, you could use whatever to keep that underneath the water. And if you have a magnifying glass so you can kind of observe more carefully, that's great. If you don't have that, that's okay too. But those are some materials that everybody should have access to at home or be able to get access to. And so then you're going to design your experiment, and you're going to be doing basically what we saw in the video, but you're going to have one of these different changes that you're going to be making, right? So maybe you'll have 4 jars, and you'll have one with a maple leaf, one with a spinach leaf, one with a lettuce leaf, and one with a leaf from a flower. And you'll expose them to the same amount of sunlight for, I mean, it says like a while, so like 20 to 30 minutes or so at least, maybe even an hour. And then you'll come back and check and see, compare, you know, the different jars. That's one example. And then you will be doing a kind of a write-up on that. So you will do your experiment or your hypothesis. And in your hypothesis, you will actually, just turn this a little bit, but you will actually be creating an if, then, because statement. And that if, then, because statement will, where you will include your independent variable in that hypothesis. So if I change the type of the leaf, then I think that this leaf will have more oxygen because more oxygen bubbles, and this is why I think that. And the because is a little loose in this situation because you're not going to have a ton of information. Maybe make some observations about why you might think that would happen. So that's part of what you need to include. And then we have the procedure. So you're going to list a step-by-step list of what you did here to test that particular variable. You can do this in a short paragraph, or you can actually do this in a step-by-step format. So it's completely up to you which kind of structure you want to use, whether that's a short paragraph or whether you want to do a list form. But again, explain what you did. And then finally, or there's 2 more steps, excuse me, create a data table to show your observations. So that data table, maybe you're counting the number of oxygen bubbles, but you need to create a data table. Don't just list your data, put this in a table form, okay? So some sort of a data table. And then finally, your conclusion. And in your conclusion, you're going to compare and contrast the control, that's what we saw on the video, with your tests. So compare and contrast the control with the tests and state whether your data supports your hypothesis, whether you need to create an alternative hypothesis and then also create that alternative hypothesis, or if your hypothesis is null and why basically you think it's null, okay? So there's 4 parts to what you need to turn into me. That's your hypothesis, your procedure, your data table, and your conclusion. And you can just put these right in here. You can insert a table in here, insert table, and you can put the number of columns and rows you want when you're putting that table in place, right? And then you can type all that information in here. This is a Google Doc, so I know that when you submit your work, you have to submit it as a Word file. When you're done with your Google Doc, just go to File and go to Download and download it as a Microsoft Word. By the way, I forgot to mention to start with, you cannot edit these Google Docs. You're going to have to make a copy of them to be able to edit them. So you can go to File, make a copy, and then you can edit that copy. Or from the beginning, before you even start, you could go to File and download as a Microsoft Word, and then you can just go ahead and have that open in Word and make your edits and stuff in Word as well. So that is the gist of what you need to do for this portfolio. Hopefully that makes sense to you. If you have any questions or concerns, you can feel free to contact me through webmail, or you can call me at school with the extension of 2204, and we can set up an appointment to work together on this. Hopefully it should be pretty simple, and I hope you have fun kind of carrying out some of these experiments. Have a great day, everybody. Take care.